Right. So, I'm Lakshman Samaranayake. I'm the editor-in-chief of the International Dental Journal, and the past dean and emeritus professor at the University of Hong Kong, as I mentioned earlier. And I have also worked as a dean in University of Queensland and also in Dubai in University of Sharjah. So my presentation, if I can just share with you. Okay, thank you very much. So my apologies about the technical issues. First time I'm using this platform. So my presentation on is the interface of oral microbiomics and new technology. And it has three parts to it. And first is oralome and the new technology. Second is new technology versus oral systemic axis. And the third is a new technology and therapeutic. So let's get on with it. I got about 45 minutes. So let's first some few definitions in this context. What is an oralome? So this is a new term which has been developed about 6-7 years ago. And some of you may not be familiar with this term. And the definition is the totality of the dynamic interactions between the ecological community of oral microbiota in the oral cavity and the host. So this is like a microbiome or just the human biome where we are living it. This is the biome where microbes within the oral cavity is living, and that is now called the oralome. And it's also called the oral ecobiome. So it's an ecological system of its own, as you will see a little bit later. So it has microbe interactions as well as host microbe interactions. And a lot of people have over periods of time done microbe interactions, but little data is available on microbe host interaction, and that's an area which needs further investigation. So you can see in this picture what it's all about. Okay. And what is more important to realize is that oralome changes. It's a very, very dynamic system. It changes from the childhood when you don't have teeth, you get a different kind of oralome as opposed to, adulthood when you have all the teeth, and then subsequently when you lose all the teeth and when you have implants and so on and so forth. And that is through the life stage. Plus, there are external factors which affect the oralome, such as the drugs you are taking, you're smoking, medications, and systemic health. So all this is very complex, leads to a very complex situation where oralome virtually changes on a daily basis. And there, within the oralome are the oral microbiota, and as you will see in a moment, and these microbial constituents change as we go along from one stage of the life to another. So what has been much interesting is the why we are knowing the revealing the depths and the mysteries of the oralome is due to this new technology, such as NGS, also called next generation sequencing. And now we also have another advanced sequencing called second generation sequencing. Then there are newer machines, which are much more faster, called Ramanomics. And then there are single cell analysis, which gives insights into how microbes behave within the interface of the human cells and the microbial cells, how they interact. So all these new advances in technology over the last 10 to 15 years have given us a totally new picture of the oralome and the oral microbiota. So this is a next generation sequencing, this new technology. It's as opposed to the cultural techniques we used to use over years, which takes years to do the oral microbiology. Now we can do it in days. Now we have another advance, another technological advance, which is called Raman spectroscopy. In fact, it is even faster than next generation sequencing. So with all these, gives us unprecedented capabilities to measure concentrations of distinct biomolecular groups in live cells as well as in microbes. And there is, for the Ramanomics, there is no sample preparation. You can take, for example, plaque samples and straight away put into the Ramanomics machines, which leads us to, gives us data within minutes. But the data, big data, we realize through these machines requires analytical processors, and these analytical processors of the massive amounts of data is in its infancy now. But then we will see in the last slide or couple of slides how the artificial intelligence is helping us to analyze this data. So with the increasing rapidity of development of artificial intelligence, we may get more and more data about our oral cavity and the oral health and oral disease. And also, so this is related to high throughput screening and automation, and this is also called lab on a chip, and so on and so forth. So just to give you a very good example of this, I'm just showing you this article from Nature magazine, which is one of the top journals in medicine and dentistry as well. And they actually studied the structure, function, and diversity of the healthy human microbiome, including the oral cavity a few years ago through the Human Microbiome Project Consortium. And this was a large group of people who got together from different parts of the world and evaluated the microbiome of, or you can see here, 4,788 specimens. Now, this was done through next generation sequencing, but it took a long time. Now you can do this in a much more rapid fashion because of the new developments, but that's a different story. So what did these guys find? What they found was that in terms of the genetic constitution, if you have a human, like we are eukaryotes, right? So the eukaryotic cells, we have only about 20 to 25,000 genes. Now the gut microbiome genetic constitution has 3.3 million genes. These are very rough guesstimates of numbers, and you can see, and it's also called the metagenome. And it's not surprising that all these organisms which are living inside the oral cavity as well as our gut, how they can influence our health as well as disease. So the more we know about these organisms, the better we can understand the processes which affect our health and disease. Now, going back to that article, what these guys found was that very interesting thing, that in different parts of the body, gastrointestinal tract, urogenital, and here you have in the yellow, orange, the oral cavity, and these are the similarity of the genetic constitution of the organisms. And there are groups of organisms living in different areas. For example, oral guys do not like to live in urogenital areas, and vice versa. So I always think this is like humans. We as humans have different creeds and different nationalities, and we tend to flock in different specific areas. Similarly, these organisms behave similarly. And in fact, not only that, but they live within communities called biofilms with very much like very community-oriented behavioral patterns. And these are some of the, now for example, this is a next generation sequencing pattern of organisms which are in, for example, in health and cancer. In cancer, for example, in health, you get the more red organisms, and in cancer types, you get the more greener type of organisms. So they have found that associated with different health and disease, there are group of organisms, and you will see this a little bit later. And once more again, you can see at the top, it's although not clear. In health, you get the red organisms predominating in this context, and in cancerous situations, you get the green organisms. So sometimes these, perhaps in the future, these green organisms can act as indicators of future carcinomas or various other cancers. Now I'm going into the oral systemic axis. So as I said, these organisms can modulate our systemic behavior or systemic health. Now this has been shown with regard to the oral cavity, particularly in the chronic periodontal disease patients. There are hundreds and hundreds of papers where they show that when there is the dysbiosis, that is the interactions between the organisms and the oral cavity changes, then these actually not only affect the local condition, but also they affect your systemic conditions as well, such as atherosclerotic vascular disease, low birth weight babies, and diabetes mellitus, and various other systemic diseases. And these three diseases which I mentioned, diabetes mellitus, atherosclerotic vascular disease, and low birth weight babies, and pregnancy complications, are the most studied up to date. Now, you will be surprised to know that there are over 220 different conditions which have been evaluated for this oral systemic axis, and how the oral cavity affects the systemic diseases. And I'll show you two more in a moment. So so we go from the big human microbiome to the oral microbiome, and then slightly diverging, going back again, I told you it's oral-ome, it's the ecosystem, and then we also have a new terminology which is called the oral microbiome. That is the totality of all organisms in the human cavity and their ecosystem. And then there is another term called the oral microbiota, and that is the bacteria, the archaea, fungi, viruses, phages, and protozoa in the human oral cavity. So I will show you in a moment how we have defined these in relation to the oral-ome and oral microbiota. So if you take the oral microbiota and the so-called oral microbiome, the microbial sphere, there is first the vast majority of the constituents which comprise the oral microbiome are the bacteria. So they are called the oral bacteriome. Okay. Then we get on to there is another group of bacteria organisms which are called archaea. So if you take the living organism, they are divided into three different groups: eukaryota, bacteria, and archaea. And this group is much less studied over the period of last 100 years, because we did not have technology to study. Now people are studying more and more of these archaea in addition to the bacteria. So these are also this is called now the oral archaeome, which is a new definition which has come into the literature. And they have found that these archaea are very primitive type of organisms which evolved later into bacteria. And these primitive organisms have specific characteristics. For example, they can live in, archaea are found in hot water springs in deep sea. They can live in plus 100 degrees, boiling water. They can live. It's a special group of organisms. And for example, they also found that in periodontal pockets, these archaea are there, and they also, the constituents can change in periodontitis and health situations. So we have here, for example, in health, you get the greener guys. This is so one of the archaea called Methanosarcina vacuolata is much more predominant in healthy gingiva than in the periodontitis gingiva. This is one example taken from this report. So that is, so we have now oral bacterium, then we have the oral archaeome, right? Then we have so these are archaea, bacteria. The third group is the eukaryotes. They are the fungi. So this is called the oral microbiome, not microbiome, microbiome. And these, like, for example, most of us listening today, we thought that like 20, 30 years ago, that oral cavity comprised organisms were just Candida only. But now we know that there are other groups of organisms within the oral cavity, such as Malassezia and various other organisms which live on our skin. And so what we don't know is how they contribute to oral disease and systemic disease. Again, another unknown area in this context. Fourthly, now we have the viruses. So there is a general understanding now. There are two major groups of viruses living in the oral cavity, and these are these viruses. The constituents are called the oral virome, and these are essentially the herpes group of viruses and the papilloma viruses. So these are the two major groups which live in the oral cavity, and I'm sure most of you have had herpetic ulcerations and so on and so forth. And this is to these viruses which live in our oral cavity as commensals, and from time to time they travel through the trigeminal nerve from the trigeminal ganglion and branches of the trigeminal nerve, and then cause this ulceration. So these are so-called resident viruses in the oral cavity. And again, they have found that there is a differentiation between periodontitis health in terms of the viral prevalence in the oral cavity. And again, done by the same group of people doing new technology in this context. And then there are smaller bacteria, bacteria-like organisms, which are much more smaller, like viral organisms, which are even smaller than viruses. They are called phages, and that is called the oral phageome. So, just skip this. And then, so you can see this is the oral microbiome is very complex. And you can see the bacteriome, archaeome, microbiome, virome, phageome comprising the oral microbiome. And this is all because of the new technology. We are seeing this. And also I have to say there are protozoa as well, such as MEB, Entamoeba gingivalis, and so on and so forth. The protozoa has not been included here, but that is another area which could be included here. So it is a very complex. And why we are seeing the complexity which is in front of us is because of the new sequencing technology and the Ramanomics and all these various technologies which has been giving us spewing new data as we speak. So very quickly we go on to the oral microbiome, which is the oral microbiome, which is going to take. So this particular oral microbiome comprises essentially of two groups of organisms living in two different lifestyles. One lifestyle is biofilms. Predominantly, most of these organisms are arranged inside communities called biofilms, and we don't have time to go into that. And that's quite a proportion suspended in saliva and so on as planktonic organisms, right? So again, I come back to the part 2, which is the new oral systemic axis, going back to that particular slide. So these bacteria cause all these major disease, not cause, associated with, sorry, I it's very, very important to realize, although we have had hundreds of papers, we could not actually show that these bacterial communities and bacterial complexes, which are pathogenic complexes, whether they cause any of these atherosclerotic vascular disease or low birth weight babies or pregnancy complications or diabetes or various other things. These are mere associations only. And one has to differentiate between association and causation. And as I say, as editor of the International Dental Journal, I get a lot of people talking about causation, but still it has not been proven. And there's a long way to go before this is to be shown. But the associations are very, very high between vascular disease and periodontitis, and vascular disease and chronic complications such as pregnancy outcomes. And what is more, there are various other diseases, so-called associations between oral cancers, pancreatic cancer, lung cancer, and esophageal cancer. And it may be that the dysbiotic oral states can cause these cancers as well, is associated with these cancers as well. So we have in the oral cavity, in the oral microbiota, we have the healthy species. On your left, you can see the good guys, and then on the right side, you get the pathogenic species. They are also called pathobionts or periodontopathogens, and there are the three major groups: Fusobacteria, P. gingivalis, then so-called actinobacter species, and so on and so forth. Then the other two conditions I just want to mention is there are associations between rheumatoid arthritis and oral microbiota, and it could be that individuals manifesting both periodontal disease and rheumatoid arthritis may suffer from the unifying underlying systemic deregulation of the inflammatory response, and the chronic inflammation may be the reason for this particular association. Then there is some very good evidence which is coming up recently and described in various journals, the association between Alzheimer's disease and periodontal disease. Particularly one of the periodontopathogens which is associated is a Porphyromonas gingivalis, and also people have to some extent sketched out the pathway by which the P. gingivalis, this is the so-called periodontal pathogen which you see in chronic periodontal disease, may cause Alzheimer's disease or may be associated with Alzheimer's disease. Okay, what are the reasons for that? The reasons are this, that for example, these P. gingivalis produce toxic proteases, and these are called gingipains, right? And this has been found in patients with Alzheimer's disease, particularly in their amyloid plaques. So one of the reasons why you get loss of memory is due to the like plaque formation in the nervous tissues, in the connecting neurons, and these amyloid plaques are thought to have been shown to contain so-called gingipains, which are derived from P. gingivalis. So you can remember these gingivalis and gingipains. It's easy to remember. So that's very, very, very just very quickly about Alzheimer's disease. Now we're coming back to new technology. How do we connect what we see today with the future of dentistry? Now, all of you have heard about personalized dentistry, I believe, and this is now becoming much more fashionable. And this is, for example, an advertisement which I taken from a Japanese paper, because of this close association between chronic periodontal disease and chronic dysbiotic oral microbiome and systemic disease, one can now say that you can analyze the bacteria in your plaque or in your oral cavity and then predict the possibility of getting oral cancers or various other diseases such as diabetes. So, beg your pardon. So this is one dental clinic. This is where they are offering this kind of services, where they sample the plaque, and from the plaque they give a personalized profile of the oral microbiota and the possibility of getting specific diseases. But this is fine to the future. Nevertheless, it's how the technology works. And these are some of the. This is another example I took from the one of the American journals, where the pardon me, is Dr. Jocker's this particular dental clinic actually saying that periodontal disease associated with atherosclerosis, respiratory problems, erectile dysfunction, diabetes, and so on and so forth, and how the periodontal how cleaning the oral cavity and having a eubiotic, so not a dysbiotic systems can lead to better health. So this is once again to do with holistic dentistry. Okay. So we are coming into the next 10 minutes or so, the new technology and therapeutics. In this context, I want to just touch on few areas: antimicrobial peptides, probiotics and prebiotics, epigenetic drugs, and matrix disruptors. Okay, let me just take you through each of these. So if you take the antimicrobial peptides, these are now, for example, if you take clohexidine or some similar drugs which are available today, when you take these clohexidine mouthwashes such as mouthwashes such as Listerine, which are made of like plant oils, what they do is they kill the bacteria non-selectively. I call it carpet bombing because it really kills everything, which leaves the good as well as the bad bacteria. So as you saw in the previous slide, there was good guys on the left and the bad guys on the right on that slide. And but these antimicrobial peptides, what are they supposed to be doing? Why are they better? Because they have selective targeting of pathogenic bacteria. So therefore, they are thought to be better than giving sort of carpet bombing through clohexidine or whatever. So one of the very highly developed examples is niacin. It's a bacteriocin like antimicrobial peptide produced by Lactococcus lactis, both active against gram-positive and gram-negative specific bacteria. They disrupt biofilms, no cytotoxicity. It retards the development of multi-species biofilms. And importantly, it has been approved by the FDA. You know how it's difficult to get approval from the FDA. So this antimicrobial peptide is now available for plaque reduction. So there are other antimicrobial peptides. I don't have time to go into these, but they are denoted as K4S4, and they have specific selective action against S mutans, AA Acinetobacterium comitas, Fusobacterium nucleatum, and then there are the other AMPs are peptoids, beta peptides, and arylamine oligomers. And now again, just like any other antibacterial, the resistance development is a challenge, and sometimes they may cause post-sensitization. Then you have nano-sized drug delivery system, number 2, as you saw, drug delivery system, DDS, as you call it. And you can see what is a nano-size. Nano-size is, just to give you an example, it's even much more. This is the size of a phage, which you saw earlier. So it's almost the size of a phage. It can go into bacteria very, very easily and kill them. The smaller the size of particles, so they are membrane-bound, small spherical particles. You can incorporate the drug into that, and once the drug goes into the system, either the organism or any other system, the outer lipid membrane dissolves, releasing the drug. So that is the nano drug delivery systems, and there are so many different delivery systems which are available currently. And for example, these particles can comprise of silver, copper, and so on and so forth, and they can release various chemicals such as quorum sensing chemicals which disrupt the biofilm, releasing the drug only at acidic pH, for example, in a karyogenic situation. So it disrupts the acidic dysbiotic biofilm, and it has maybe has potential for caries reduction. So so they have found that a nano DDS system it kills biofilms 4 times more effective than a free drug because it's very targeted and very selective. Okay. They have reduced both the number and severity of carious lesions in vivo compared to the free drug, which is the farnesol they used in the nano drug delivery systems. So, and also the important thing is that these nano drugs are these drugs inside these vesicles are very sensitive to degradative enzymes. So therefore, these nanoparticles actually protects the drug until they reach the target. So these are the important things in the nano drug delivery systems. Then all of you have heard about probiotics. Now, now here I'm talking about number 3, which is the prebiotic. And it's a natural or synthetic food ingredient or supplement that modulate the microbiome to benefit the host. So I call them, actually you can call them the lunch for good bacteria, as it were, it's just a slang thing. So it's actually you're nurturing the good bacteria such as Lactobacillus reuteria, Lactobacillus acidophilus, which are good for both for the oral cavity as well as systemically. So the common prebiotics are arginine nitrate and monosamines, and these can be given in containers. And remember, these are not probiotics, these are prebiotics. These are chemicals which feed the good bacteria and does not nourish the bad bacteria. So, so as I said, these are lunch for good bacteria. They are arginine, nitrate, and monosamines. They have different ways of activity. Arginine prevents caries by buffering the acids. Nitrates useful against both caries and periodontitis by increased ammonium production and increasing the pH also. And then monosamine, selectively triggers the growth of commensal oral bacteria. Okay, but there are a lot of additional studies, much more research is required in this context. And then we have probiotics. All of you know about probiotics. And they are live microbes which, administered in adequate amounts, confer a health benefit for the host. And for example, Lactobacilli and Streptococci have antibacterial activity against all these periodontopathic bacteria. And I don't want to go into detail because you know all about it. Because even if you got some kind of diarrhea, you go to the doctor. Nowadays, they give Lactobacilli suspensions. So a lot of studies. We have done a recent study, meta-analysis showing that gingivitis and periodontitis can be reduced overall by these probiotics. And then it has pro-inflammatory effects, modulates the pro-inflammatory effect. It reduces inflammation. And then there's chewing gum with Lactobacillus reuterii, which one study led to reduction in gingivitis, some example. And then there is another example where Bacillus subtilis reduce the bone loss and attachment loss. Then another thing which I want to show is another probiotic which directly affect caries is an organism which called Streptococcus dentisani. Denti means the teeth, and sani means sanitary. So that's how they call it, dentisani. And when you have in taking Streptococcus dentisani in some kind of compound into the oral cavity, it straight away goes into the carious areas where there is this pH, low pH, right? And it has been shown to increase the pH and thereby reducing the karyogenicity. That has been reported in Frontiers in Oral Microbiology. And then there are some notions that where you can get, for example, saliva from healthy donors and then purify it and then give it to situations where you have chronic infections such as candidiasis in immunocompromised patients as oral rinsers. And so that eubiosis, this is a good bacteria given to a bad bacterial state, dysbiotic state, and goes back to good bacterial state. So these are some of the new technologies which may come into the future, which you may see in the future. So there are some drawbacks of probiotics because the number of probiotic cells needed for so you need large amounts of bacteria. You need to transportation in viable form because they once given in, they have to grow inside the oral cavity or in the gut. Yeah, so therefore people are talking about micro encapsulation of probiotics, such as the nano drug delivery systems as well. So this is whatever it is. And we have now finally a last few slides, extracellular polymeric material disruptors. So some of you may have, I'm sure all of you know that the oral biofilm has a very protective outer sheath, which is called the extracellular polymeric matrix, which is which has which really protects the microorganisms living inside the matrix. It's like a big, like a bomb shelter. You know, nowadays we are talking about bomb shelter where incoming bacteria cannot get in, incoming like even chemicals and antibiotics cannot get in due to the thick exopolysaccharide they have. So if you can disrupt, so the theory is that if you can disrupt this matrix of the oral biofilm, then indeed, then that you can reach your targeted bacteria. So there are two strategies for this. First is to disruption of the synthesis of the extracellular polysaccharide, and then disrupting the secretion and targeting its composition and structure once it's formed. So before it's formed and after it's formed, you can actually destroy the matrix. So these are what people are working on this context. So disrupting EPS synthesis and secretion has been achieved by different kinds of compounds in different researchers like Karaholis and so on. Composition and structure has been disruption has been achieved by using some of these very innocuous enzymes such as dextrinases, mutanases, and dispersing, and so on and so forth. Finally, we come on to the epidrugs, so-called epidrugs. So these are drugs that inhibit or modulate disease-associated epigenetic proteins. And so the examples are bromodomain and so on. And one example in an animal model is this particular bromodomain ameliorates or reduces gingival inflammation and alveolar bone destruction in P. gingivalis induced experimental periodontitis. So people don't know why exactly the mechanisms behind this, and these are called epigenetic drugs that inhibit or modulate the proteins which are derived from proteins produced by these bacteria. So I'm coming on to the last slide here. So artificial intelligence, all of us, all people are talking about AI and how it's going to change our behavior and every aspect of our behavior. And similarly, the new technology can be impacted by artificial intelligence. So hopefully, we through the help of AI, we should be able to find a solution for caries and periodontal disease. The other day, somebody asked me, he said, Professor, you have been working on these two diseases for the last 60, 70 years, but you haven't got any, very big results like curing COVID-19 or vaccine, or we don't have anything of that sort. So with hopefully with the rapid AI analysis of this big data reservoirs on drugs, we have together with this new technology, through Ramanomics or NGS or next generation NGS, we are generating so much data, it's almost impossible for humans to analyze it. So if you feed this into the AI system with its multiple analytical processors, hopefully they will produce some kind of possible new drugs or new drug compositions which can be used against periodontal disease or caries or various other oral diseases. And they can pinpoint targeted drugs. And this is called to some extent called in silico finding of new drugs, where the drugs are, because all this is previously, lots of these chemicals have to be screened, hundreds of chemicals have to be screened, and tested one by one for the effect on a specific disease. Now, in silico, within the computer, you can test all these drugs for disease-causing organisms and how they are affecting their membranes and how their enzymes can be affected by the use of these new drugs. So in silico, finding of these new drugs will hopefully, through artificial intelligence, we should be able to analyze these new data and hopefully get some new drugs in the future. And then, of course, tailoring drug therapy, personalized care is another area which I'm not going to go in just now. There's no time. So this is where the new technology actually really plays a part, because as you know, when you give a drug, the same drug will have different effects on different people. It will work on some people. There's a minority, it will not work. But if you have personalized care, where as you saw earlier, where you can do the personalized analysis of your microbiome or the oral cavity, and if you know whether the person is susceptible to any disease, and if it yields more accurate data, then of course it will be much more useful than current carpet type of drug therapy. So this is a very, very complex situation, and this last slide shows you how all these new drug delivery systems and all these prebiotics, probiotics, and all this can affect the oral microbiome and possibly bring about the development of a good microbiome and leading to better health, as you can see on the left side. Together with, of course, there are other factors which influence better health. But I believe that oral eubiosis, where there's good microbiome and a good oralome ecosystem, this could lead to better health and better compliance, can also lead to better health through appropriate interventions. So last but not least, I would like to thank you for being here. And also I would like to thank again the Dr. Marian Constantinescu for the kind invitation, and for me to organize this symposium, and everybody else. So thank you very much.
Many thanks for your very interesting lectures, Sam.
Yeah, thank you so much.
Thanks.